Welcome back to the deep dive. This is where we try to pull out the really key bits of knowledge, make you genuinely well informed. And today we are jumping right into the fascinating world of plant shoots. That means basically everything you see above ground: stems, leaves, eventually flowers. Our mission for this deep dive is to really explore the incredible story of how that shoot gets built, how it grows, and how all its parts connect. We're drawing some pretty amazing insights from Raven biology of plants. When you look at a plant, right, the shoot is doing a lot of the heavy lifting, literally. Stems, they're all about support and transport, getting those leaves up into the sun. And leaves, well, they're the powerhouses, aren't they? Making food through photosynthesis. But the way it all actually comes together down at the cellular level, it's way more intricate than you might think. Okay, let's unpack this.
Right. So to really get the shoot, you got to start right at the very tip, the shoot apical meristem. And it's not, just a simple growth zone. It's more like a dynamic control center. It's constantly adding cells to the plant body. And at the same time, it's repeatedly producing these tiny little leaf primordia. Those are the very beginnings of leaves and bud primordia, which those become the lateral shoots, the branches.
So it's like a little factory churning out parts.
Exactly. A very precise, ongoing process. It's creating these fundamental building blocks. What's interesting, too, is how it stacks up against the root. The root apical meristem has that tough root cap for protection.
Right, pushing through the soil.
Yeah. But the shoot meristem doesn't have that. It's much more delicate, so it's often shielded by these young leaves that are kind of folded over.
They're little bodyguards.
Sort of, yeah. And we should clarify a term here. We often say shoot apex, but technically, the apical meristem is just the very, very tip above the youngest leaf bit. The term shoot apex actually includes that meristem plus the slightly lower region, the subapical part, where those young leaves are just starting to take shape.
Okay, so a subtle but important distinction. The peak versus the whole summit area.
Precisely.
So at this tiny, absolutely vital tip, how is it organized? Are there specific layers or zones that determine what cells become what?
Oh, absolutely. It's a really elegant system, particularly in most flowering plants. We call it a tunica-corpus organization. Think of the meristem as having distinct layers. The outermost layer, or sometimes layers plural, that's the tunica. Now, these tunica cells, they primarily divide anticlinally.
Anticlinally, okay. What does that mean exactly?
Imagine slicing a cake straight down, perpendicular to the surface. That's anticlinal division. It increases the surface area without adding more layers, like stretching a sheet wider.
Gotcha. Expanding outwards.
Right. Then beneath the tunica, you have the corpus. This is a body of cells that divides in, various directions. And crucially, it includes periclinal divisions. Those are parallel to the surface.
Okay, so if anticlinal is slicing down, periclinal is like slicing horizontally through the cake, adding layers.
Exactly. That's what adds bulk and thickness.
Yeah.
It's like inflating the meristem from within. But here's something really remarkable. A cell's ultimate job, its fate, isn't strictly determined by which cell it came from. Its lineage.
Really?
Yeah. It's much more about where it ends up in the developing structure, its final position.
Wow, so location, 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 even for plant cells, that's quite different from how we often think about development.
It gives the plant incredible flexibility, this developmental plasticity. It allows it to adapt its form based on all sorts of cues, internal and external.
Fascinating. Okay, you mentioned position is key. You also mentioned different zones within this meristem structure earlier.
Yes. Within this tunica-corpus setup, we can identify 3 functional zones. There's a central zone. It's a bit less active in terms of cell division, acts kind of like a reservoir of initial cells or stem cells.
Like a reserve pool?
Kind of. And surrounding that is the peripheral zone. This is a highly active ring of cells. It's busy generating the sides of the stem and, importantly, the beginnings of the leaves.
Okay.
And then below the central zone is the pith meristem, which contributes to the core, the pith of the stem. And what's truly amazing is that this whole operation is under really sophisticated genetic control. We've learned a massive amount from studying model plants like Arabidopsis.
Right, the lab rat of the plant world.
Pretty much. We found genes that act like on switches, like SHOOTMERISTEMLESS or STM, which gets the meristem started, and another called WUSCHEL or WUS, which is crucial for maintaining those initial cells. Then you have other genes like the CLAVATA genes, CLV1, 2, and 3, that act more like off switches or regulators. They kind of rein in the WUS activity to keep the meristem size in check.
So it's a balancing act, a genetic feedback loop.
Yes, exactly. WS promotes the initial cells, CLV dampens it down. It keeps the whole population of initial cells perfectly balanced for continuous controlled growth. It's really elegant. And these zones, they directly give rise to the primary tissues that make up the mature stem. So the outermost tunica layer, the L1, that develops into the protoderm, which becomes the epidermis, the skin of the plant.
Makes sense. Outer layer becomes outer layer.
Right. And the peripheral zone that contributes to the procambium, that's the precursor to the vascular tissues, xylem and phloem, and also part of the ground meristem, which forms the cortex and sometimes part of the pith.
Okay.
And the pith meristem, well, that forms the rest of the ground tissue, mostly the central pith.
It's just incredible that this tiny region, this microscopic zone, orchestrates the entire above-ground structure of the plant. Mind-blowing. But, okay, once these basic tissues are laid down, how does the stem actually, get longer? How does it gain height?
Ah, good question. That primarily happens through internodal elongation. See, unlike roots, where you have pretty distinct zones for cell division, elongation, and maturation all lined up.
Yeah, we talked about that with roots.
Right. Stems are a bit different. Growth isn't quite so spatially separated. Elongation happens mainly between the nodes, those points where the leaves attach. Initially, when new leaves are forming rapidly up at the apex, the nodes are really close together. You can barely see the internodes, the spaces between them.
Okay.
But then, those internodal regions start to stretch, quite significantly in many cases. And that pushes the nodes apart and pushes the whole stem upwards. This elongation can happen in several internodes at once.
So it's like a telescope extending,
Yeah.
stretching out the bits between the leaves.
That's a pretty good analogy, yeah.
Mm-hmm.
And in some plants, especially grasses, there's another mechanism too. They have intercalary meristems.
Intercalary, meaning in between.
Exactly. These are localized regions of meristematic activity, usually found at the base of the internodes, or sometimes near the base of the leaf itself.
Yeah.
They can keep dividing and adding cells even after the main apical meristem has moved way up. It allows for continued elongation lower down the stem.
Ah. Is that why grass keeps growing back after you mow it?
That's a big part of it, yes. Those intercalary meristems keep pushing out new growth from the base.
Clever stuff. So the basic architecture gets built, then it gets stretched into place, sometimes with these extra growth zones. But even with this fundamental growth pattern, you look around and stems look different. Inside too, right? What determines their internal plumbing and structure?
That's a crucial point. While the basic job is similar support, transport the internal organization of the primary vascular tissues, the xylem and phloem, shows quite a bit of diversity. We can roughly group them into three basic types.
Okay, 3 types. What's the first one?
First type, you see this in plants like, say, basswood or tilia. The vascular tissues are arranged in what looks like a more or less continuous cylinder. If you were to cut across a young stem, you'd see the outer epidermis, then a cortex region, then this ring of vascular tissue, and finally a central pith.
A solid ring of plumbing?
It appears quite solid, yeah.
Yeah.
But if you look really closely, it's technically composed of vascular bundles that are just separated by very, very narrow regions of ground tissue. These are called interfascicular regions. But visually, it often looks like one continuous vascular cylinder. Very strong design.
Okay, continuous cylinder, type 1. What's type 2?
Type 2 is where you have a cylinder, but it's made of discrete vascular bundles. They're clearly separated by wider regions of ground tissue, which we often call pith rays or interfascicular regions. Think of plants like elderberry or alfalfa or even buttercup.
So like separate pipes arranged in a circle with packing material between them.
That's a good way to picture it, yeah. In these bundles, you typically see the phloem carrying the sugars developing on the outer side toward the cortex, and the xylem carrying water develops on the inner side towards the pith. This arrangement allows for more flexibility, perhaps, and is common in plants that might undergo secondary growth later.
Right, where they add wood.
Exactly.
Yeah.
Though some herbaceous plants with this structure, like buttercup, have closed bundles, meaning they lose their potential for secondary growth early on.
Okay, so discrete bundles in a ring. Got it. What's the third type?
The third type is characteristic of most monocots. Think grasses, corn, lilies, palms. Here, the vascular bundles appear scattered throughout the ground tissue.
Scattered, not in a ring.
Nope. If you cut a corn stem, for example, you'll see bundles dotted all over the place. There's no clear distinction between a cortex and a pith region. The whole thing is just ground tissue with vascular bundles embedded in it.
Huh, that seems messy. Or is there a reason for it?
It's actually highly efficient for rapid growth, which many monocots do. These scattered bundles are also typically closed, like the buttercup ones, meaning no secondary growth.
Yeah.
And they're often surrounded by a tough sheath of sclerenchyma cells for extra support.
Wow. Here's where it gets really interesting. Such different internal blueprints for doing similar jobs. Support and transport solved in multiple ways. But okay, the stem isn't an island. It's the support structure, the highway for the leaves. How does this internal plumbing, whichever type it is, connect seamlessly to those leaves?
Yeah, that connection is fundamental. That's why we often use the term shoot.
Yeah.
It really emphasizes this tight physical and developmental link between the stem and its leaves. The vascular system for a leaf, it doesn't just connect up after the leaf forms. The procambial strands, the precursors to the vascular tissue, actually start differentiating below where the tiny leaf primordium is forming.
So the connection starts before the leaf really even exists.
Exactly. It differentiates upwards, growing into the developing leaf, ensuring there's vascular continuity right from the very beginning. It's not an afterthought, it's integrated development. We call the vascular bundles that diverge from the stem's main vascular system to enter a leaf traces.
Leaf traces, okay.
And where a leaf trace departs from the vascular cylinder in the stem, it often leaves behind a little region of ground tissue in the cylinder just above where the trace branched off. That's called a leaf trace gap.
A gap. Does that weaken the stem?
Not really a hole, more like a region where parenchyma tissue interrupts the vascular cylinder. It's particularly noticeable in stems with that continuodylander or discrete bundles in a ring. In scattered bundles, it's less obvious. And botanists even have a term for a single stem vascular bundle plus all the leaf traces associated with it as you go up the stem. Symposium. It just highlights how interconnected this whole system is. It's one continuous vascular network throughout the entire shoot.
A symposium.
Okay.
It really paints a picture of an integrated system, like a complex highway network with exits for every leaf. And speaking of patterns and design, the way leaves are arranged on the stem, that's often incredibly regular and beautiful. What can you tell us about those arrangements? How do plants manage that precision?
That arrangement is called phyllotaxis. And yes, it's often remarkably precise and mathematical. It's another testament to the plant's developmental control systems. You see several common patterns. There's spiral or helical phyllotaxis, where you get one leaf per node and they spiral up the stem. Think of an oak or a mulberry tree.
Right, winding upwards.
And then there's opposite phyllotaxis, where leaves arise in pairs at each node, directly across from each other. Maples are a classic example. If successive pairs are at right angles to the previous pair, we call that decussate, like in coleus.
Okay, pairs.
And you can also have whorled phyllotaxis, where 3 or more leaves emerge from a single node. Culver's root is a good example of that.
So how do these patterns form? It seems too precise to be random.
It definitely isn't random. Scientists have been puzzling over this for ages. Early ideas involve things like the first available space, a new leaf pops up wherever there's room, or an inhibitory field idea, where existing young leaves somehow prevent new ones from forming too close.
Makes intuitive sense.
It does. But the mechanism was unclear. More recently, biophysical forces were proposed, focusing on stresses and strains in the surface layers of the meristem. But the current leading hypothesis, the one with the most evidence now, is the auxin-based model.
Auxin again. That hormone seems to be everywhere in plant development.
It really is crucial. The idea is that high concentrations of auxin signal the spot where a new leaf primordium should initiate. Specialized transporter proteins, called PIM proteins, actively direct auxin flow to create these high concentration peaks.
Okay, so auxin marks the spot.
Right. And then once a young primordium starts to develop, it acts as a sink for auxin, drawing the hormone towards itself.
Ah, so it uses up the local auxin?
Exactly. And by doing that, it lowers the auxin concentration in the immediate vicinity, effectively creating that inhibitory field we talked about earlier. It prevents another leaf from forming right next door.
So the next peak of auxin, and thus the next leaf, will form in the next available spot farther away.
Precisely. This dynamic interplay between auxin transport creating peaks and young primordia acting as sinks can generate all those regular patterns, spiral, opposite, world, depending on the specific dynamics of auxin flow and meristem geometry. It's a self-organizing system based on hormone transcode.
That is really elegant. A chemical signal creating geometric perfection. Amazing. Okay, let's shift from the patterns on the stem to the leaves themselves. They come in just a staggering variety of shapes and sizes. What are the basic building blocks of a leaf, and how do they vary so much?
You're right, the diversity is incredible. But most leaves share some basic parts. There's usually a broad, flat part called the blade or lamina. That's the main photosynthetic surface. And often there's a stalk, the petiole, which attaches the blade to the stem.
Blade and petiole. Okay.
Some leaves also have little appendages at the base of the petiole, called stipules. They can vary a lot, from tiny scales to leaf-like structures. And some leaves lack a petiole altogether. They're called sessile. Their blade attaches directly to the stem.
Like in many grasses.
Exactly. In grasses and some other plants, the base of the leaf often expands into a sheath that wraps around the stem for a bit. Grasses also have that little flap, the ligule, where the blade meets the sheath.
Right. Now, what about leaves that look like they're made of lots of little leaves, compound leaves?
Yes, that's a major distinction. A simple leaf has an undivided blade, although it might be lobed or deeply cut. A compound leaf has a blade that's divided all the way down to the central vein, forming distinct segments called leaflets.
So multiple leaflets make up one compound leaf.
Correct. And there are different types. Pinnately compound leaves have leaflets arranged along a central axis, the rhiches, kind of like a feather. Think of ash or walnut trees. Palmately compound leaves have leaflets that all diverge from a single point at the tip of the petiole, like fingers from a palm. Horse chestnut is a good example.
How can you be sure if you're looking at a small leaf or just a leaflet from a bigger compound leaf?
The key diagnostic feature is the bud. True leaves, whether simple or compound, will always have an axillary bud in their axil, the angle where the petiole meets the stem. Individual leaflets never have buds in their axils. Also, all the leaflets of a single compound leaf generally lie in the same plane, whereas individual simple leaves on a branch can orient themselves in various directions towards the light.
That's a really useful tip. Look for the bud in the axil.
Always works. Now, beyond these basic forms, leaf structure is hugely influenced by the environment, especially water availability.
Right, you mentioned xerophytes and hydrophytes.
Exactly. Xerophytes, plants adapted to arid conditions, go to great lengths to conserve water. They often have very thick cuticles, stomata, the pores for gas exchange, that are sunken into pits or grooves, maybe dense coverings of hairs or scales, all to reduce water loss.
And hydrophytes, the water plants.
Their challenges are different. If they're submerged, they might lack stomata entirely, absorbing gases directly from the water. Floating leaves usually have stomata only on the upper surface, the one exposed to air. Their internal structure might have large air spaces for buoyancy, too.
Makes sense. Adaptations for their specific water situation. What about the inside of a typical leaf, say, from a moderate environment?
Okay, a typical mesophyte leaf. Inside the epidermis, you have the mesophyll. That's the ground tissue specialized for photosynthesis. It's usually packed with chloroplasts. And critically, the mesophyll has lots of intercellular air spaces. These are connected to the outside world via the stomata, allowing for efficient gas exchange, CO2 coming in, oxygen going out.
Right, fuel in, exhaust out.
Pretty much. In many broadleaf plants, the mesophyll is differentiated into two layers. An upper layer, the palisade parenchyma, consists of tightly packed, column-shaped cells. They're oriented perfectly to capture sunlight hitting the top of the leaf.
Maximum light harvesting.
Exactly. Below that is the spongy parenchyma. These cells are more irregularly shaped, and there are much larger air spaces between them. This facilitates the gas movement deeper inside the leaf.
So palisade for light capture, spongy for gas exchange.
That's the general idea, yes. Though some plants, like corn, have mesophyll that isn't clearly differentiated into these two layers. And running through this mesophyll, of course, are the vascular bundles, or veins.
Leaf's own plumbing network.
Right, continuous with the stem's vascular system via those leaf traces we discussed. The pattern of veins varies too. Most non-monocot flowering plants have netted venation, or reticulate venation, a branching network, often with a prominent midvein, and progressively smaller branching veins.
Like a ring map.
Yeah. Whereas most monocots, like grasses, typically show parallel venation. Numerous veins run parallel along the length of the blade, interconnected by smaller transverse veins.
Like railway tracks.
Good analogy. Within each vein, the xylem, water transport, is usually located on the upper side towards the top of the leaf, and the phloem, sugar transport, is on the lower side. The larger veins are mainly for transport into and out of the leaf, while the very fine, minor veins are embedded within the mesophyll and are crucial for collecting the sugars produced during photosynthesis.
Loading up the sugars for export.
Precisely. And these smaller veins are often enclosed by one or more layers of compactly arranged cells called the bundle sheath.
Bundle sheath, does that do anything special?
Yes. It plays a critical role in regulating the movement of substances between the mesophyll and the vascular tissue. Think of it as a checkpoint. Sugars have to be actively loaded through the bundle sheath cells to get into the phloem. Some bundle sheath cells even have Casparian strips, like the endodermis in roots, controlling water flow.
Wow, another layer of control. Plants are just full of checks and balances.
You really are. And sometimes extensions from these bundle sheaths reach all the way to the upper and lower epidermis, providing extra support to the leaf structure.
So much detail in a single leaf.
And the adaptability. Remember that aquatic plant, Calitris heterophylla?
Yeah, the one that makes different leaves underwater versus in the air.
Right. Research showed that hormones like gibberellic acid could induce the water-form leaves, mainly by affecting cell wall properties and allowing cells to expand more under turgor pressure. Abscisic acid, another hormone, promoted the land-form leaves. It really suggests that the physical pressure within the cells, the turgor, interacting with wall properties plays a huge role in determining the final shape and size of the leaf. The plant can literally adjust its form based on internal pressure responding to external conditions.
That's just a fantastic example of plasticity, a plant literally changing shape to fit its world. It really makes you think about specialization, like in grasses. What makes grass leaves stand out structurally?
Grass leaves definitely have some unique features, especially when you compare C3 and C4 grasses, which have different photosynthetic pathways.
C3 and C4,
Right.
related to efficiency in different climates.
Exactly. C4 grasses, like corn or sugarcane, exhibit something called Krantz anatomy. Krantz means wreath in German,
A wreath, why?
because if you look at a cross-section of a C4 leaf vein, you see the large bundle sheath cells forming a ring or wreath around the vascular tissue. And then the mesophyll cells are often radially arranged around that bundle sheath. It forms two distinct concentric layers involved in their specialized photosynthesis.
Okay, Krantz anatomy for C4. What about C3 grasses, like wheat or rice?
They lack that clear concentric arrangement. Their bundle sheath cells are generally smaller, contain fewer chloroplasts, and sometimes there's even an additional inner sheath layer. The distance between veins is also typically greater in C3 grasses compared to C4 grasses.
Interesting structural differences linked directly to function.
Absolutely.
Yeah.
And another thing you often see in grass leaves are bulliform cells. These are groups of large, thin-walled cells usually located on the upper epidermis.
Bulliform cells, what do they do?
The leading hypothesis is that they act as motor cells. When the plant starts to lose too much water, these cells lose turgor faster than others, causing the leaf blade to fold or roll inwards along longitudinal lines.
Ah, a water-saving mechanism,
Right.
reducing the exposed surface area.
Precisely. A very clever adaptation for surviving dry spells. What's fascinating here is you see how these anatomical details, Krantz anatomy, bulliform cells, vein spacing, all directly impact how the grass functions, how efficiently it photosynthesizes or conserves water. Structure and function perfectly intertwined.
It really is. So how does a leaf actually come into being, from that tiny primordium at the meristem?
Well, it all starts with a small group of founder cells,
Mm-hmm.
maybe just a handful, maybe up to 100, right there in the peripheral zone of the shoot apical meristem. These cells start dividing and expanding differently from their neighbors. The first visible find is usually a slight bulge on the side of the apex called a leaf buttress.
Oh, buttress, okay.
This buttress then grows outwards and upwards, forming the young leaf primordium. As it develops, it usually becomes flattened on its upper side. The broad blade of the leaf is then typically formed by the activity of marginal meristems, zones of cell division running along the edges of the young primordium.
So growth along the edges makes the flat blade.
Largely, yes. The duration and pattern of activity in these marginal meristems play a big role in determining the final leaf shape. Short activity leads to simple, smooth-edged leaves. More prolonged or differential activity can create lobes, teeth, or even compound leaflets. But a lot of the leaf's growth, especially its expansion, happens through intercalary growth. That means cell division, and importantly cell enlargement, occurring throughout the developing blade, not just at the margins or tip.
So it grows from within, too.
Very much so. And unlike the shoot apex, which has indeterminate growth and can potentially grow forever, leaves exhibit determinate growth. They grow to a specific size and shape, and then they stop. Growth typically ceases first at the tip and progresses towards the base.
A pre-programmed final form.
Essentially, yes. And the vascular system develops in a very coordinated way, too. In broadleaves, the main midvein procambium develops upwards from the stem connection. Major veins branch off and develop outwards. But the finest network, the minor veins, actually develops in the opposite direction, starting near the leaf tip and differentiating downwards or backwards towards the base, connecting up with the larger veins as they go.
Backwards from the tip. Why?
It ensures that the leaf tip, which is often the first part to fully expand and begin serious photosynthesis, is the first part to get fully hooked up to the vascular network for exporting those sugars.
Clever again, always thinking ahead.
Plants are masters of logistics. Monocot leaves, like grasses, develop a bit differently, often starting as a hood-like structure that encircles the apex, with growth zones concentrated more towards the base.
So what does this all mean? How does this development process allow leaves to adapt so well, like those sun and shade leaves you mentioned?
Right, that plasticity is key. Whether a leaf develops as a sun leaf or a shade leaf depends heavily on the light conditions it experiences during its growth. Sun leaves, growing in high light, end up being smaller but thicker. That thickness comes primarily from developing more extensive palisade parenchyma, sometimes 2 or even 3 layers deep. They also tend to have more stomata and a denser network of veins.
Built for high performance and bright light.
Exactly. They have a much higher maximum rate of photosynthesis when light is abundant. Shade leaves, on the other hand, grow larger and thinner. They maximize surface area to capture the limited light available. They usually have only a single layer of palisade cells, often with more chlorophyll per cell.
Optimized for low light capture.
Right. But they can't handle highlight very well. They get damaged easily. The amazing thing is that the same plant can often produce both types of leaves, depending on where they are on the plant. Sun leaves at the top, shade leaves lower down.
So the plant actively adjusts its leaf factory based on local conditions. Remarkable.
It's a fantastic example of developmental plasticity, adapting the plant to its specific microenvironment.
Okay, so leaves develop, they function, they adapt. But eventually, their time comes. Deciduous trees drop their leaves, but even evergreens replace them over time. How does a plant manage to shed a leaf cleanly without leaving itself vulnerable?
That programmed shedding process is called abscission. It's a very controlled, orderly process, not just the leaf randomly falling off. It happens at a specific location, the abscission zone,
Yeah.
usually right at the base of the petiole. Structural changes occur here well before the leaf actually drops.
What kind of changes?
2 distinct layers typically form within this zone. There's a separation layer, composed of cells with weak walls that are destined to break down. And beneath that, closer to the stem, a protective layer forms. The cells here become heavily impregnated with suberin and lignin waterproof, decay-resistant materials.
Like forming a scab before the wound even happens.
That's an excellent analogy. Before separation, the plant salvages valuable mobile nutrients from the aging leaf. Things like magnesium, nitrogen, and amino acids, sugars pulling them back into the stem for reuse. Resource conservation.
Smart, don't throw away the good stuff.
Exactly. Then enzymes get to work in the separation layer. They specifically target the middle lamella, the pectin-rich glue holding cells together, and sometimes even the cellulose in the primary walls. The cells essentially just fall apart from each other. Once this separation layer breaks down, the leaf is only held on by the vascular strands, the veins. A little wind or even just its own weight is enough to make it detach.
And the protective layer is already there.
Yes, the protective layer is fully formed before the leaf falls. So when the leaf detaches, it leaves behind a clean leaf scar on the stem, which is actually the surface of this protective layer. It effectively seals the wound immediately, preventing water loss and pathogen entry. It's incredibly well orchestrated.
It really is a very neat and tidy process. Now, we've talked about the shoot stem and leaves and how they connect, but how does the shoot's vascular system link up with the root system down below? They seem quite different structurally.
That is a really fundamental connection point for the whole plant. And you're right, the typical arrangement in the root, often a solid vascular cylinder with xylem and phloem in alternating radii, is quite different from the stem, which usually has vascular bundles surrounding a central pith, with phloem on the outside and xylem on the inside of each bundle.
So how does the plant bridge that gap?
It happens in what's called a transition region. This usually occurs very low down in the hypocotyl, the part of the stem below the cotyledons, or sometimes spanning the hypocotyl and the upper part of the root. It's not an abrupt switch. Instead, there's a gradual reorganization. The vascular strands twist, branch, fuse, and reorient themselves. The xylem poles from the root might fork and rotate outwards, while phloem strands shift position, eventually forming the stem's arrangement of bundles around a newly formed pith.
Wow, that sounds incredibly complex geometrically.
It is very complex, and the exact pattern varies between different plant groups. But the crucial outcome is vascular continuity. The plumbing remains connected throughout, ensuring water and minerals from the root can reach the shoot, and sugars from the shoot can reach the root. It's a masterpiece of developmental engineering that happens very early in the seedling's life.
A seamless transition despite the different architectures. Amazing! That connection really sets the stage for the plant's entire life. But eventually, for many plants, vegetative growth gives way to something else, the flower. How does that transformation happen?
The transition to flowering. That's a profound shift. The development of a flower, or an inflorescence, which is a cluster of flowers, usually marks the end of vegetative growth for that particular shoot apex.
So the apical meristem changes its job completely.
It does. It converts from a vegetative apex to a reproductive apex. This involves significant physiological and structural changes, often triggered by environmental cues like day length or temperature or internal developmental signals. A key difference is that the reproductive apex typically shows determinate growth. Unlike the vegetative apex that can potentially grow indefinitely, the floral meristem is programmed to produce a specific set of organs, the floral parts, and then its activity ceases.
Limited growth for a specific purpose.
Exactly. Often, just before flowering, you might see changes like rapid internode elongation, pushing the developing flower buds up. The apex itself often broadens and becomes more dome-shaped, gearing up for producing the floral organs. And those floral organs, the sepals, petals, stamens, and carpels, are generally considered to be evolutionarily modified leaves. They arise in successive whorls on the floral meristem, typically in that order. Sepals first, then petals, stamens, and finally carpels in the center.
Modified leaves. That makes sense, given they arise from the same kind of meristem.
Right. And understanding how the meristem knows which organ to make in which whorl has been a huge area of research, especially using genetics. You might have heard of homeotic mutations in flowers, where organs develop in the wrong place, like getting petals where stamens should be, creating double flowers.
Yeah, I've seen those.
Well, studying those mutations, particularly in Arabidopsis, led to the development of the famous ABC model of floral organ identity, now often expanded to the ABCDE model.
The ABCDE model. Okay, sounds like developmental building blocks.
It basically is. It proposes that different classes of genes, let's call them A, B, C, D, and E genes, are active in overlapping domains, or whorls, within the developing flower. Their combined activity specifies what organ develops where.
Can you give an example?
Sure. According to the model, in the outermost whorl, only A and E genes are active, and that combination specifies sepals. In the second whorl, A, B, and E genes are active together, specifying petals.
Okay.
In the third whorl, it's B, C, and E genes, which specifies stamens. And in the centermost whorl, C and E genes alone specify carpels. The D genes are involved mainly within the carpel, specifying ovule development, working with C and E.
So it's the unique combination of active gene classes in each whorl that dictates the identity.
Precisely. And the E genes are interesting. They seem to be required for any of the A, B, or C functions to actually happen.
Yeah.
If you knock out the E function, all the floral organs tend to develop as leaf-like structures.
Wow. So E is like a master switch for floralness.
Kind of, yes. And their interactions, too, like A and C functions, tend to inhibit each other. It's this complex combinatorial code, controlled by transcription factors encoded by these genes, that orchestrates the whole process. There's even a key gene called LEEY that helps switch the meristem from vegetative to floral in the first place, turning on the ABC genes. This raises an important question. Just how remarkable is this intricate genetic control that creates such diverse and beautiful flowers from a basic ground plan?
It truly is remarkable, such precise control leading to such variety. And speaking of variety and modifications, plants don't just modify leaves into flowers. Stems and leaves get adapted for all sorts of other surprising jobs, right?
Oh, absolutely. Plants are incredibly resourceful inventors when it comes to modifying their basic parts. Think about climbing plants. Many use tendrils for support.
Tendrils, yeah. Those curly things.
Right. And those tendrils can be modified stems, like in grapes. Or they can be modified leaves or parts of leaves, like the terminal leaflets of a garden pea becoming tendrils. Some stem tendrils, like in Boston ivy, even develop adhesive discs at the tips to cling to surfaces.
Clever. What else?
Sometimes stems become flattened and leaf-like, taking over the main role of photosynthesis. These are called cladophylls or phylloclades. Asparagus is a good example. Those feathery green leaves are actually modified stems. Many cacti are another example, where the fleshy green body is the stem.
How do you know it's a stem and not a leaf?
Remember the rule about buds. True leaves have buds in their axils. Cladophylls, being stems, might bear flowers or leaves, but they won't have a bud in their axil where they attach to the main stem.
Got it. The bud rule holds. What about sharp, pointy things? Spines, thorns, prickles?
All modifications for defense, but with different origins. Spines are technically modified leaves like on a cactus. All those spines are leaves that have become hard, sharp, and non-photosynthetic. Thorns, on the other hand, are modified branches. They arise in the axils of leaves, where a branch would normally form, but they develop as sharp, woody points instead. Hawthorns have true thorns.
Okay, spines are leaves, thorns are branches. What about roses? They have thorns.
Ah, technically roses have prickles. Prickles aren't modified leaves or stems. They're just sharp outgrowths from the epidermis and cortex tissue of the stem. They're more superficial. Think of them like hardened hairs almost.
Spines, thorns, prickles. Good distinctions to know.
Definitely. And then there's storage. Stems and leaves are often massively modified for storing food or water.
Like potatoes.
Exactly. The Irish potato is a tuber, which is a swollen underground stem tip specialized for storing starch. Those eyes on a potato, those are the nodes, each containing dormant axillary buds.
And the potato is a stem, not a root.
Correct. Compare that to a bulb, like an onion. A bulb is essentially a very large bud. It has a small conical stem at the base, but most of the bulb consists of fleshy, modified leaves packed with stored food.
Stem versus leaves as the main storage part.
Right. And then there's a corm, like in gladiolus or crocus. A corm looks similar to a bulb from the outside, but internally it's mostly thickened, fleshy stem tissue storing food, with only thin, papery scale leaves on the outside.
Tuber, bulb, corm, all underground storage, but different parts modified.
Precisely. And sometimes it's above-ground stems, like the fleshy stem of kohlrabi, or even petioles like in celery or rhubarb, that become specialized for storage.
Okay.
And don't forget water storage, especially in succulent plants from arid regions. Cacti store water primarily in their fleshy stems, but many other succulents like aloe, agave, or stone crops have incredibly thick, fleshy leaves adapted for holding large amounts of water in specialized parenchyma tissue.
An amazing array of adaptations. And just briefly, we should mention the really extreme leaf modifications, like in carnivorous plants.
Absolutely. Pitcher plants, sundews, Venus flytraps. Their leaves have evolved into incredibly sophisticated traps to capture insects. They don't do it for energy, primarily, but to obtain essential nutrients like nitrogen and phosphorus, which are often scarce in the boggy soils where they live. It just highlights the extraordinary evolutionary potential residing in the basic structure of a leaf.
What an incredible deep dive this has been into the plant shoot. Seriously.
Yeah.
We've gone from the very tip, that dynamic shoot apical meristem, and unpacked its tunica-corpus organization and that really elegant genetic symphony controlling its growth. We looked at the different ways stems are built inside those primary structures, each a unique solution for support and transport. And we saw how deeply connected the stem is to every single leaf it bears. We marveled at the sheer orderliness of leaf arrangement, phyllotaxis, driven by that key hormone, auxin, and discovered just how profoundly leaves adapt their internal structure, even their overall shape, to suit their environment, like those sun and shade leaves. And of course, the huge transition to flowering, that genetic ABCDE code, plus all those amazing ways stems and leaves get repurposed as tendrils, spines, tubers, bulbs. Incredible ingenuity.
It really is. If we connect this to the bigger picture, what truly stands out, I think, is the incredible precision combined with the adaptability of plant growth. You have these microscopic cell divisions, these intricate genetic feedback loops, all leading to this vast array of plant forms we see around us. Every structure feels like a testament to millions of years of, well, evolutionary fine-tuning. It's a system built for both reliable, robust growth, and yet also for flexible response to whatever the environment throws at it. That's how they've managed to thrive almost everywhere on Earth.
Yeah, this deep dive genuinely makes you look at every tree, every weed, every blade of grass with a whole new level of awe, doesn't it?
Yeah.
So thinking about all this, I'll leave you, our listeners, with this thought to mull over. How does this deep fundamental understanding of primary plant development, how stems and leaves are built and function, how does this knowledge continue to shape our approaches to agriculture, to conservation, especially now as we face new environmental challenges and increasingly look to plants for sustainable solutions and innovations? Something to think about.